Cellular respiration occurs within engines called mitochondria. The mitochondria is shaped like a pill capsule, and it is composed of two membranes, one surrounding the other. The inner membrane is folded in many different dimensions in order to maximize surface area. The space in between the two membranes is called the intermembrane space. The space inside of the inner membrane is called the mitochondrial matrix. Embedded within the inner mitochondrial membrane is a group of proteins known as the electron transport chain, ETC, and another one called the F1F0 ATPase. There are so many of both types of these membrane proteins that the inner mitochondrial membrane looks like grass-covered ground. Here's how it works. Normal state. Pull respiration. An electron from the Krebs cycle, which takes place in the center of the mitochondria, the mitochondrial matrix, inside of both the inner and outer mitochondrial membranes, is deposited into the first protein of the ETC, which is sticking out into the matrix. That electron is very high in energy. Once it gets deposited into the top of the ETC, it gets sucked out of the matrix towards the intermembrane space by the next protein in the ETC, because that protein has slightly more affinity for the electron. That higher affinity forms a temporary bond between the electron and the protein, releasing a small amount of energy in the process. That energy is then used to pump protons from the matrix into the intermembrane space, the space between the inner and outer mitochondrial membranes. That causes a higher concentration of protons to form in the intermembrane space than in the matrix. That concentration forms a gradient of positively charged particles. We are talking about charged particles, and a gradient of charged particles is by definition an electrical field. That electrical field is then dissipated by protons fluxing through the ATPase, producing ATP, the energy currency of life, as a byproduct. At the very end of the electron transport chain, a familiar friend is waiting: oxygen. Oxygen is the final oxidizing agent in the electron transport chain. It combines with the electron permanently, forming water. All along this chain of membrane proteins, oxygen is gently pulling energy out of the food we eat in the form of high-energy electrons in motion. Let's refer to this whole process as pull respiration. The pull of oxygen's attraction for high-energy electrons is what fuels life. Acute stroke. Failure of cellular respiration. In the setting of a stroke, a very delicate, highly energy-intensive organ suddenly becomes starved of oxygen. Now what? There isn't as much oxygen around, so there is less pulling taking place through the electron transport chain. Since there is less pulling, there are less electrons moving, and they are moving slower. That means fewer protons getting pulled from the matrix into the intermembrane space by the work of oxygen via the electron transport chain. That means a weaker gradient of protons fluxing from the intermembrane space back into the matrix via the ATPase. That means markedly lower energy, ATP, production. Famine is established. Cells are at risk of destruction because life is highly energy dependent. Pull respiration is fully dependent on oxygen delivery to tissues. Acute stroke plus pulsed magnetism, push respiration. Push respiration, on the other hand, is an entirely new concept. It does not require oxygen delivery. An electrical gradient is supplied across a tissue. A TMS pulse does this. When a portion of the electrical gradient vector aligns with the axis of an ATPase embedded in an inner mitochondrial membrane, that electrical gradient facilitates proton flux through the ATPase. The ATPase does not run in reverse, so the component of the electrical vector in line with the ATPase's proton vector facilitates proton flux through the ATPase, producing more ATP and adding to the cell's energy.